and we're very fortunate to have the discoverer of the most recent of the possible associated infectious agents, Dr. Judy Mikovits, talk to us next. She began her career at the Cancer Institute, getting her degree from George Washington, and established a research program at the Cancer Institute for many years. In Dennis's terms, you might call her seasoned, like me. And from there, she went to the Whittemore Peterson Institute and has primarily pursued the work on XMRV. And most recently, she's the director of research there, but is concentrating on translational research. Judy?
I guess in Tony's terms, I'm actually doing reverse translational research director, so we'll have to change that again. Thank you very much. So I'm going to talk today, obviously, about XMRV and MLV-related viruses in CFS. So I want to start by asking, basically, what do we know about XMRV? Well, the demonstration by Sam Chow in Bob Silverman's lab of integration sites by the group in Emory Petros group of in situ fluorescence, in situ hybridization in chromosomes in unmanipulated tissue, and the demonstration in our laboratory of antibody responses, we know that XMRV is a human infection. And we know that although it's closely related to xenotropic, polytropic, and murine leukemia viruses, XMRV is not an endogenous retrovirus, either in humans or in mice. How XMRV got into humans is unclear. But it's interesting, Steve Goff and Bob Silverman has shown its expression is stimulated by androgens and inflammation, and that's important in the discussion in talking about cortisol. So cortisol directly stimulates the LTR, this virus. So we'll look at the schematic organization of the virus on the next slide. I'll just first say that this is a classic EM of a gamma retrovirus. So XMRV is the first human gamma retrovirus, and of the human retroviruses now we have HIV, HTLV1, and now XMRV. These are complex retroviruses because they encode accessory proteins that regulate their expression, whereas XMRV encodes only the structural GAG, polymerase, and envelope genes, and the expression of the virus is regulated by the LTR. So it's cis-acting elements is the on-off switch. So hormones and particularly cortisol and androgens can actually be an on-off switch for this virus. So how do we detect human retroviruses? We detect retroviruses clinically by serology, detection of antibodies to the viral proteins in the blood, by Western blot, that is doing blots to detect viral proteins that are expressed in cells, by looking at the provirus, the integrated provirus by DNA PCR, by RT-PCR, reverse transcriptase PCR, to look at the virion-associated RNA. There are two copies in a retrovirus of RNA, and you can also look at partial transcript using RT-PCR. Finally, you do culture the virus on indicator cell lines or fresh from tissue and detect the virus after incubation of those cell lines with blood cells or plasma. So we use this classical approach to do the first direct isolation of XMRV from the blood of CFS patients. And I show you two parts of that. So first this is a single round DNA PCR done by Bob Silverman and Joy Dasgupta. And what we see here is the GAG and the envelope DNA located, could be proviral, not expressed DNA, in patient cells with CFS. Interestingly, patient 1118 here we see is negative by DNA PCR, but when we did cell-free transmission from the plasma of that patient, cells from the same plasma that was taken on this day, we could see by Western blot for both envelope and GAG, we could see high levels of virus by Western blot actually had been transmitted to those cells. And this is really the first suggestion that there was some variability in the sequence, as we had only 6 full-length isolates in the gene bank, and they were very highly similar. So this suggested there was variation in this sequence of these viruses, and they weren't as similar as we expected. And that hypothesis was confirmed in our work, confirmed and extended by Lowe and Alter late last year, and they used the same nested methods that we used in Lombardi et al., which were different from the single round methods, and they found GAG sequences that were more closely related to polytropic and modified polytropic murine leukemia viruses than to XMRV. Importantly, they found 86% of the CFS patient samples they used from a cohort described by Tony Komaroff. And it's important there because he diagnoses patients very similar to Dan Peterson as those severely, largely homebound or bedbound patients. So they found those GAG sequences in 6.8% of the control samples, which would be 20 million Americans who could potentially harbor this virus. Interestingly, 7 of 8 of the patients, when they went back 15 years later when they did this work and got fresh blood, they found the same polytropic and modified polytropic MLV-related viruses. They rigorously controlled their studies and found no contamination of mouse DNA, either by mitochondrial DNA or IAP particles. So we wanted to, taking these together, we wanted to go back and do more extensive look at some of the samples in patients that were both negative and positive in our study. And we looked then at the envelope region. The envelope primers of Urisman et al., the Silverman primers, would not detect polytropic and modified polytropic viruses. So Kathy Jones made primers, designed primers that went from the propol all the way down into the envelope, and these would detect the polytropic, the modified polytropic, as well as XMRV. And what she did was she did individual single-cell cloning from a single patient. This patient was one of the full-length clones in the database, showing XMRV. She does a lot of buttons, so I'll do the buttons first. So you could see here that XMRV is clearly in this patient and compares to the database sequences. But they also cloned another sequence of envelope out of this patient, and you see here that it is more closely related to the modified polytropic and polytropic sequences described by Lowenalter in their paper. So when you look, just to show the similarities, there are big differences between XMRV and polytropic sequences. If you look at the primers that were used in many of the replication studies or the studies, and those by Urisman et al. And in the single round PCR, you see that there were enough differences shown in the colored region that you would never see the polytropic or modified polytropics using these PCRs that were largely used in the negative studies, just looking at that GAG region. So it's important then we conclude from these studies that several individuals carry both the XMRV and polytropic murine leukemia related viral sequences as confirmed by individual cloning and sequencing of the PCR products. So Kathy went on to do a large number of the patients from our original work and a phylogenetic analysis of those, where you see in green all of the endogenous retroviruses. In yellow are the VP62-like viruses shown here. The XMRV-like, the polytropic are in green, and modified polytropic. And you see throughout here the WPI samples showing many of these patients have polytropic sequences as well as the described XMRV. There's another MULV-related, it's called actually xenotropic MULV-related virus that's in a B lymphoblastoid cell line. It's a human cell line, and not much is known about the virus, but it's full-length sequences. But it's interested in thinking about the B-cell tumors. The reason I went to the Whittemore-Peterson Institute was the description by Dan Peterson of the large number of chronic lymphocytic leukemia and mantle cell lymphoma in that population. So we show variation in XMRV sequences and then low levels of the replicating virus, which I won't be able to show you. There's a lot of editing, ApoBec editing, we know, that actually keeps infectious virus. So there are only low levels of replicating virus in the blood and low levels because it may be tissue-oriented, as we heard earlier. So those are some of the reasons why groups may fail to detect it. Therefore, we look for other footprints of XMRV infection in unmanipulated plasma. So the first